Hello, my name is Eric Padilla, and I will be, doing a video presentation on two case studies. So we have one case study number 2, which has to do with a 16-year-old female who has been diagnosed with muscle deficiency in muscle glycogen phosphorylase. And then we have case study number 4, which has to do with an infant who has some issues maintaining her blood sugar, especially during the fasting period. Has some hepatomegaly going on, so we'll dive further into that later on in this presentation. So just to kind of kick things off, let's go ahead and jump into the first case study, which has to do with our 16-year-old female who's deficient in glycogen muscle phosphorylase. So her first question is regarding how glycogen is used during exercise. So basically, just to kind of start things off from just from the basics. So when we eat food, our body breaks it down into smaller molecules. So for example, carbohydrates are broken down into glucose, or also known as sugar. The body can either use this glucose through a process called glycolysis, or to generate energy, or it can kind of store it for later, in the form of glycogen. So there's two places that glycogen is stored at, which is in the liver and the skeletal muscles in our body. So during exercise, you're causing your muscles to work, and therefore they need more energy in the form of ATP. And to kind of generate more ATP, the body needs more glucose to create it. And one way to get that glucose, again, is if it's stored in the form of a glycogen. So in order to break down that glycogen, we need that muscle glycogen phosphorylase to break it down. So again, so that we can make that available, or to that can be used to generate energy for the muscles. Since she doesn't have this enzyme, she can't break down glycogen. One way to think of it is, again, a good analogy is kind of a warehouse full of glycogen, kind of in our liver or skeletal muscles, and then we have our workers, which is our enzyme, the glycogen muscle phosphorylase. So the enzyme is responsible for moving sugar in and out of that warehouse. However, if we don't have workers, then that warehouse just kind of that's just has glycogen stored in there, but nothing's happening with it. So that's her situation in regards to what's going on with her diagnosis. The second question she has is she likes to take long walks, so she can still do that. She just has to be aware of kind of take some precautionary measures, as well as be aware of some things. So she'll get some exercise intolerance kind of in the first few minutes of exercise. This is because, again, her body can't immediately break down that glycogen. So she may experience some muscle cramping, muscle fatigue. It will eventually resolve in about 10 minutes with rest or continuation of exercise. This is because your body is resorting to alternate pathways to make glucose available for your muscles, so that it can be used to generate energy to be utilized for muscle contraction. One recommendation is, drink a sugary drink or a meal 40 minutes prior. This is because, glucose will be more readily available, as opposed to, kind of storing it for later. She just has to be a little bit careful, because, too intense of a workout or anaerobic workout can cause some skeletal muscle damage, cause kidney failure down the road due to rhabdomyolysis. Third question she has, will she have to maintain her blood sugars? No, she will not have to maintain her blood sugars, because, again, for one, the liver is responsible for maintaining blood sugars. So when when you have low blood sugar, your body has mechanisms in place to, like, hey, I'm low on blood sugar, hey, releases glucagon, and that basically breaks down the glycogen in the liver, eventually to glucose, which is then let out into the bloodstream to maintain blood sugar, as well as deliver glucose to your skeletal muscles, or your brain, or your red blood cells. So skeletal muscles do not play a role in maintaining blood sugars. And then last one is, will she have any issues with excessive lactate production? She's pretty smart for a 16-year-old, so for her, she doesn't have to worry about that again, because of that she can't break down glycogen effectively, so there's not as much lactate being produced because of that deficiency. In addition, since the lactate that's produced from glycolysis can be transported to liver, where it's then converted into pyruvate, and then that pyruvate can then eventually be turned into glucose to make to supply or to be delivered to the skeletal muscle, so I can use it to make energy. All right, moving on to our next case study is case study number 4. Excuse me. And so with this individual, there's a couple of diagnoses that are on the table for her. Main one of them is, or there's 4 of them here, but based on her symptoms, for her the diagnosis would be glycogen storage disease, in particular. So individuals, with this disease, also known as von Gurk, these individuals are unable to break down glycogen into glucose all the way, because they lack the enzyme glucose 6 phosphatase, which is responsible for the dephosphorylation of glucose 6 phosphate into glucose. Therefore, we're not making that transition to glucose. We were unable to maintain those blood sugars. So it makes sense for her during fasting states, where she is unable to break down that glycogen all the way down to glucose, thus resulting in hypoglycemia and those jittery spells for her. In addition, the hepatomegaly is due to that buildup of glycogen in her liver. Blood lactate is increased because of the epinephrine administration. So what epinephrine does in the body is basically, again, trigger the liver to break down glycogen. So for her, she can't break down glycogen all the way down to glucose. Therefore, despite the administrative epinephrine, there's no increase in blood sugar because she's lacking that glucose 6 phosphatase enzyme. However, she does increase in blood lactate. This is because, as glycogen is being broken down partially to glucose 6 phosphate, but that glucose 6 phosphate then accumulates due to the absence of glucose 6 phosphatase. Since glucose 6 phosphate is accumulating, that basically means more glucose 6 phosphate can enter glycolysis, and due to glycolysis, more pyruvate is generated. This overloads the body, or overloads like the TCA cycle, and as a result, the body turns to alternative methods to, on what to do with that pyruvate. So one of the ways is through lactase dehydrogenase to convert pyruvate into lactate. So that explains the spike in her blood lactate levels. As far as the other 3 diagnoses of hereditary fructose intolerance, galactosemia, and glucose 6 phosphate dehydrogenase deficiency, she does not have any of those. Mainly if she did have hereditary fructose intolerance, for one, breast milk does not have fructose in it. The primary sugar is lactose, and that's made up of galactose and glucose. So in individuals with this hereditary fructose intolerance, they lack an enzyme called aldolase B, which cleaves fructose 1-phosphate into glyceraldehyde and dihydroxytone phosphate. There wouldn't be, for her, that kind of rules that out of the table because of those little steps there. Again, she's able to digest the galactose and from the milk. So that kind of bridges off to the second point, is the galactosemia. So again, she's able to metabolize galactose from the lactose in the breast milk. We wouldn't have seen this like 2 months down the road. We would have pretty much seen this when she's at birth. As you know, so she would have had symptoms such as jaundice, vomiting, impaired growth, because in individuals with galactosemia, they lack an enzyme called galactose 1-phosphate uridyl transferase, which basically, helps break down that galactose 1-phosphate into glucose 1-phosphate, which can again eventually be turned into just glucose down the road. And then lastly, the patient does not have G6-PDH or galactose 6-phosphate dehydrogenase deficiency, because this enzyme is an important enzyme in the pentose phosphate pathway, and it helps create NADPH. So that NADPH plays a role in helping keep glutathione in a reduced state. Reduced glutathione helps oxidize hydrogen peroxide in red blood cells through a enzyme called glutathione peroxidase. Accumulation of hydrogen peroxide can be very damaging to the cell membrane, cause some oxidative damage. So it's important to have that enzyme, so we're not lysing our cells and causing hemolytic anemia. So that kind of just, for her, again, diagnosis, to summarize things, diagnosis for her is that glycogen storage disease. So again, she's not able to break down that glycogen all the way down to glucose, and as a result, she's having some issues maintaining her blood sugar, that hepatomegaly, as well as, again, no response from the epinephrine as regards to, increasing her blood sugar, and, also increasing her lactate level, because, again, she's not breaking that glycogen all the way down. So that's wraps up my presentation regarding the case studies. So yeah, thank you.